So I'm Mike Coburn, Executive Vice President of Research America, and I want to thank you for joining us for our next panel discussion, Can New Discoveries Combat Antimicrobial Resistance? The panel will discuss some of the newest research tools to help answer this question. We're delighted to have our moderator with us today, Rachel Roubein, who is the National Healthcare Reporter and author of The Health 202 with the Washington Post, Michael Craig, Director of the Antimicrobial Resistance Coordination and Strategy Unit at the Centers for Disease Control and Prevention, Dr. Amy Mathers, Associate Professor of Medicine and Pathology at the University of Virginia, Dr. Jeff Miller, the Fred Kavli Chair in NanoSystems Sciences and Director of the California NanoSystems Institute and Professor of the Department of Microbiology, Immunology, and Molecular Genetics at UCLA, and Dr. Mike Ybarra, Vice President of Advocacy and Strategic Alliances and Chief Medical Affairs at PhRMA. You can find their bios in the toolbar at the bottom of the virtual environment. And you can type your questions in the Q&A box, and the panel will try to get to those throughout the conversation. So it is a pleasure, Rachel, to turn the program over to you.
Hi, thanks everybody for being here. Thanks to the panelists and the audience listening at home or in the office or wherever you are. Excited to moderate this conversation. And as mentioned, if you have any questions and answers as you think of them, just pop them in the chat and I'll try and get to them. So I really wanted to start and kind of set the stage here for antimicrobial resistance. Over the past 2 years, almost 80% of patients hospitalized with COVID-19 received an antibiotic from March to October 2020. And I'm wondering, Michael Craig, is that why hospitalization-related infections grew from 15% from 2019 to 2020? Were there other factors?
Thanks, and a great question, and very excited to be on the panel today. I think the challenge that we saw with the COVID-19 pandemic certainly had to do with a lot of different factors. I think the chief one that we saw that most affected antimicrobial resistance was that we had just a huge influx of sicker patients going into hospitals. And in hospitals, you do see the transmission of some of our most dangerous antimicrobial resistant pathogens. And with that influx of sicker patients into those healthcare settings, with those healthcare settings being overwhelmed with the pandemic, we saw a greater transmission of those pathogens. Certainly a contributing factor to that is additional antibiotic use, and that additional antibiotic use can potentially make a patient more susceptible to getting an infection, can disrupt their microbiome, and can have other impacts on them. So all of this sort of confluence and constellation of things we think really had a detrimental impact at reversing a lot of the progress we saw in preventing antimicrobial resistance before the pandemic.
And I'm wondering a little bit, Dr. Amy Mathers, in terms of this, is this something that you've heard of when you talk to people in hospitals? Do they need to kind of retrain staff on some of this infection control? What's kind of the happening sort of on the ground in facilities here?
Yeah, so as a director of an antimicrobial stewardship program, during the pandemic, our resources from a pharmacy standpoint and from a physician standpoint were stretched really thin. And so there was some knowledge that had been accumulated and some progress and programs in how to judiciously use antimicrobials. And some of that knowledge was lost, I think, through training and through disruption in training. And then the fact that, like Dr. Ybarra or Dr. Craig just mentioned, there was such a disruption in practices, and we kind of had all hands on deck to control the pandemic and deal with those patients, that there was just some institutional knowledge lost. And so I think that that's been one of the really big challenges, that we need to go back to basic principles and retrain on some of the judicious antibiotic usage that we know is so important to making sure that we're not further selecting for antimicrobial resistance when we give antibiotics to patients.
Dr. Michael Ybarra, what did you see on the ground in hospitals? You also work as an emergency physician, and what are some of the best practices, with Amy mentioning that's something that needs to be retrained. So what are some of the best practices?
Yeah, well, thanks for the question and thanks for having me. It's great to be with so many experts. I think you hit on all the right points. You know, we'd previously seen a decline in drug-resistant infections, but we're now seeing an increase of around 15% year over year since the pandemic began. And hospital-acquired infections are becoming alarmingly more resistant. We're seeing resistance patterns in Acinetobacter and Candida, which is a fungus, and multi-drug resistant Pseudomonas. And so, as you mentioned, I'm an emergency physician by training. I also work in industry, and this issue of antimicrobial resistance is really, really important because we're seeing so many patients come in. Treating infection is a really critical part of both aspects of my job. So I think a key thing that we need to do kind of as a community is make sure that we're following evidence-based guidelines when it comes to COVID-19. So if you're seeing a COVID-19 patient, there are treatments available for those patients, thanks to the hard work of industry in collaboration with government to bring antivirals to market that are safe and effective in the appropriate treatments. We're still seeing a lot of co-prescribing of antibiotics, unfortunately, for a viral infection. Early on in the pandemic, a lot of patients were ventilated. They needed mechanical ventilation. They were put on a ventilator when they had severe respiratory infections. And we saw a lot of hospital-acquired infections in those settings. We've sort of moved past that at this point. And so I think at this point, following evidence-based guidelines when it comes to treating COVID-19 with antivirals is really, really important.
And what is sort of from the CDC's perspective, the guidance that they're giving providers right now in terms of administering antibiotics for patients?
Yeah, and I think Mike hit on the high points of it, but I think from our perspective, it is getting back to the first principles of infection prevention and antibiotic stewardship. So it is really making sure that you have the appropriate, you're prescribing the appropriate medicine to that person at the right time and the right scenario. And as for a COVID-19 patient, we have alternatives now, and that's the great opportunity, and really one of the successes of the pandemic was getting so many of those innovative products to market so quickly, so that we have those things to treat these patients with, and that we're not relying upon things that we weren't sure about. And one of the things that folks relied upon the most was antibiotics. I think the other thing that we're stressing from the CDC perspective is getting back to infection control. Noting that when people are having procedures in hospitals, we have very meticulous infection control processes that need to be followed. And under the pandemic, because of the stretching of the staff and the influx of sick patients, not all of those things could be done the way they needed to be done. And we need to get back to making sure that we are doing everything that we can as healthcare providers to take care of those patients as safely as possible.
And Dr. Jeff Miller, wanted to bring you in. What sort of impact have you been seeing from just kind of the research perspective on COVID-19? How has this sort of shaped your thinking on the landscape of AMR?
Yeah, thank you, Rachel. There have been a lot of impacts. Certainly the rapid development of point-of-care diagnostics, and also the incredible activity in terms of drug development is very impressive and important. It sets a precedent. Also, we've seen the sort of genesis in terms of public health of a really interesting vaccine modality, an mRNA-based vaccine with nanoparticles. And probably the most important feature of that is it can be reprogrammed. It can be programmed to keep up with evolution. We haven't necessarily done as good of a job of doing that as we could, but it's certainly a remarkable development in vaccinology and a real improvement. Of course, we also need to work on the acceptance problem, which is a separate issue. And there have been other sort of consequences as well. There's been an upgrading of biosafety level training capacity. More people are interested in infectious disease research. And hopefully we'll be seeing the effects of this on other viral diseases as well as bacterial diseases and other pathogens.
Now, Michael, the CDC has a national action plan for combating antibiotic-resistant bacteria. Can you give us a brief overview of that plan and what's needed to fulfill its goals, what the status is of actually completing these 5 points of action?
Absolutely. Thanks, Rachel. So would would note that it's actually not just CDC, but it's actually all of the U.S. government works under the Combating Antibiotic Resistant Bacteria, or CARB action plan. And this is the National Action Plan of the United States that's intended to address the problem of antimicrobial resistance in our country and also globally. So there are 5 areas that cross-cut the U.S. government. They go into surveillance, prevention, drug development, diagnostic development, alternative development for things like vaccines or other preventatives. And then they also go into what do we need to do globally. And all of those areas, I would just note, we've made tremendous progress on since the inception of the National Action Plan in 2015, but there's so much more that we need to do. And I think the challenge that we see is that, as was noted, is that COVID has really taken us backwards as a country in terms of progress. So as CDC reported in July, we had 15% increase in infections and deaths from AR infections. And, that really erased years of progress that we had seen prior to the pandemic. And I think what we would really note, and I think what the administration is trying to do, is reinvigorate and jumpstart our action on antimicrobial resistance. There is a lot of work that needs to be done. We have a second iteration of our action plan that was released in 2020. And really, there's a lot of work that needs to be done in terms of resourcing that action plan and supporting all of the agency's efforts to make further progress here. Because I think the thing that we note is that we need to fund and support these things now so that we're not in a place with the next pandemic of trying to fight twin pandemics of an emerging infection like COVID, as well as sort of the endemic issues of antimicrobial resistance that we deal with every day.
And to drill down on that a little bit, you mentioned reinvigorating this. You mentioned resources. Are you talking more funding from Capitol Hill, from Congress? What kind of specifically do you think is needed to jumpstart this?
Yeah, and absolutely more resources from Congress are needed. And in fact, in the president's budget this year, there were additional resource requests for antimicrobial resistance. I know specifically to CDC, the pandemic preparedness plan also had additional resources for antimicrobial resistance development. And the administration has put forward ideas on how we can also help support the antibiotic drug pipeline and trying to make sure that that is being prioritized as well, so that we have antibiotics available whenever we need them.
Michael Ybarra, Michael had mentioned the antibiotic drug pipeline. Can you talk to us a little bit about where the industry is in terms of drug development here?
Yeah, absolutely. It's a great question. And just again, to sort of make it really personal, infection is one of the most common conditions that I treat in the emergency department. So whether it's somebody that cut their leg in the Potomac when they were, either kayaking or whatever water activity, or it's somebody that has, a catheter and develops a multi-drug resistant UTI. These things are out there. And I think, you don't realize it until it impacts you or it impacts your family. But we are seeing more and more drug resistant infections. And unfortunately, the pipeline is relatively light for new antibiotic development. And that's why I appreciate the comments of Michael, Amy, and Jeff of, more work needs to be done here to support the development. Unfortunately, there have been some high profile bankruptcies of antibiotic development companies recently that I think underscore the challenges of developing medicines to combat antimicrobial resistance. And a couple of reasons for that. Number one, it takes a long time to develop these medicines. I think there is this perception that we were able to develop antivirals and vaccines so quickly. Why can't we just do that for AMR? Well, in part because we need that kind of collaborative focus to do so. And secondly, another big component of what I say sort of as a physician is if a new antibiotic comes to market, I won't be able to prescribe it sort of on purpose, right? You don't want the emergency physician to be taking that brand new antibiotic and prescribing it widely, which means the market for the company that developed it is relatively small. So that's why solutions are needed, because you don't want antibiotics to come out and be prescribed in the same way that we've used vaccines. Vaccines for COVID are to prevent infection. If a new antibiotic comes out to treat the multi-drug resistant infection, you sort of want to save it until you absolutely need it. So that's sort of the setup for it. There is a unique sort of ecosystem in place to help the development of new antibiotics. That includes everything that's been mentioned to date. There's also CARB-X. We'll talk more about the AMR Action Fund that industry stood up And then the incentives piece, which is, and one potential solution is the Pasteur Act, which creates sort of a subscription model, almost like Netflix, you can think of it as. So you're not being paid based on volume, but instead sort of a subscription that the government could pay to create a market in this really unique situation. So that's sort of our high level approach. I know we're going to dig in on a lot of this in the coming few minutes, but that's sort of the overview of where we're at from an industry standpoint.
And Dr. Jeff Miller, what are the challenges that you see here? And can you talk to us a little bit, you have experience in precision antibiotics. Can you talk to us a little bit about the status and the landscape of that as well?
Sure, thank you. And just to bounce off of what Mike Ybarra just mentioned, 80% or so of the antibiotics that are currently in the clinical pipeline come from small companies. Many come from the biotech sphere, small innovative companies interested in developing antibiotics. As big pharma has pulled out of the antibiotics business, this has a chilling effect and sort of a trickle-down effect to where the innovation lies. And much of the innovation lies at that sort of academic startup company entrepreneurial interface. And when big pharma is no longer developing, producing, doing clinical trials, et cetera, and acquiring these companies, it becomes very difficult to get investment for the downstream. And so basically we're leaving a public good, an essential public good, up to the pressures of the marketplace. And that's a very precarious position to be in. And I know we'll talk more about the Patriot Act, et cetera. And this is really important. In terms of other challenges to producing antibiotics, we'll talk about these a little bit later, but they stem from permeability characteristics of bacteria, the extreme distance between different bacteria in terms of the relationships, and overall need to make antibiotics are not only efficacious, but also safe. We use small molecule antibiotics at concentrations that are much higher than most other drugs that we take. So the development of these life-saving therapeutics is in a fairly precarious place. You asked about precision. We can talk about this a little bit later when we talk about phage, but one of the things that we see are that antibiotic usage does have an effect on the protective microbes that inhabit us. Our gastrointestinal tract has 200, 300 different species, trillions of bacteria, and they protect us from invasion by competitors, in many cases pathogens. And when we take antibiotics, there is great evidence that that barrier is compromised. And in fact, it degrades over time. We pass our antibiotics, our microbiomes on to offspring during natural childbirth, for example, from mother to child. And so there's a progressive degradation that many of us are concerned about in terms of the health of our beneficial microbes. And then finally, when we use broad host range antibiotics that are effective against many different pathogens, many different bacteria, we impose selection for transmission of resistance to any bacterium that's susceptible. And so in our microbiomes, we all have antibiotic resistance determinants that are sitting there waiting to be spread to other pathogens. So precision is one way to deal with both of these issues, but there are some requirements that we don't yet have to make that approach efficacious.
And just off of what you said, I wanted to bring Michael Ybarra and has that been sort of the case where this is something that smaller companies and that big pharmaceutical companies aren't looking at developing these antimicrobial drugs, or are not kind of putting resources towards that?
Well, I think what it's more is that there is sort of a, we have to admit that there's a bit of a market failure here because of the unique situations that I mentioned before. Again, if a new antibiotic comes to market, it's going to be sort of saved for those worst case situations. You don't want, again, like the emergency physician like me prescribing the brand new antibiotic for the, wound infection until you know for sure it's multi-drug resistant or it's a super sick patient that's in the hospital. So I think it just recognizes that this is not the same situation situation as what we're sort of used to with many other diseases, where there is sort of a market and there's volume associated with that. So there are a number of companies that are investing, but I think what we admit is that there's not enough given the challenge of and the public health threat that it faces. There's a lot of really exciting science happening, but we need the sort of long-term investment to create a more sustainable funding situation for this very challenging field. So, one idea that was mentioned is that PhRMA Act, that it sort of intends to incentivize companies to develop novel antibiotics, and they are truly highly novel antimicrobial agents, and to ensure that sort of companies make it through the valley of death, as we like to call it. You've got those early ideas, those early molecules that maybe are in lab, and you want to kind of carry them through so that they don't fail and they ultimately do make it to patients. So a lot of work to do. There are companies in the space, both big and small, but I think we all recognize that there's not enough given the threat that this kind of crisis poses to the public.
And just real quick, before we move on here, on the PhRMA Act that you both had mentioned, now that's a bipartisan bill from Senators Michael Bennet and from Senator Todd Young. PhRMA has come out in support of that bill. Is that something you guys are talking to lawmakers on Capitol Hill? And what do you see as the prospects for the legislation this year?
That's a good question. You're right. There's actually a pretty broad coalition of folks that are supportive of it, including the infectious disease physicians. I think it's one solution, and it's a really important one. And so we sort of view it as needing to drive comprehensive policy reforms like the PhRMA Act. You mentioned it's bipartisan, which is very helpful. It's also bicameral, so it's in the House and the Senate. So we're hopeful. We're going to do what we can to get it done as soon as possible, because again, it's going to take a long time to bring these antibiotics to market. So it's not like flipping a switch. We need to put these incentives in place so we can have a more sustainable market and allow new drugs to come to market over time.
Dr. Mathers, I saw you give a thumbs up. What are your thoughts?
Yeah. So as somebody who's in the Infectious Disease Society of America, we've been talking to congressional leaders and trying to educate and just make sure everybody gets their questions answered, and we think thoughtfully and bring this forward as a group, because again, as an infectious disease physician, I do see the other side of it where you end up with these untreatable infections, and it can be just devastating. You just don't have an antibiotic to treat people with. And so we definitely need movement, and we need additional drugs for patients, both in precision, which I think is a great idea and approach for all the reasons that Jeff mentioned, but also probably just standard drug development as well, and making sure that those companies are supported to get through so that we've got product for patients. And I, as a leader of a stewardship group, would just really second what Dr. Ybarra is saying about the fact that I'm going to restrict those drugs as soon as they get to market. And we do restrict those drugs because if we use them, we know with a lot of the new novel agents that have come to market in the last 5 years, resistance development is much faster than it was, say, 10, 20 years ago, for a lot of really complex reasons. But some of the agents just aren't quite as durable, mostly because the antibiotic resistant bacteria have an arsenal of genes of drug resistance by the time they're getting into a hospitalized patient in the modern era.
And Dr. Miller, you'd mentioned phage therapy, so I wanted to turn to that. So phage therapy, which uses bacteriophage viruses to treat bacterial infection. In March 2021, the NIH awarded the first ever grants to study phage therapy. So I'm wondering, Jeff, what are the questions researchers still need to answer about this treatment? And this is still experimental, so how common is the usage of phage therapy right now?
Boy, I'll be interested in Dr. Craig's take on this as well. So bacteriophage, just in terms of introduction, are viruses that infect bacteria. They infect a bacterial cell, they replicate in that cell, they burst out of the cell, killing it. And usually one phage goes in, one virus goes in, and maybe 50 or 100 will come out. And so they amplify themselves very rapidly when they kill their bacterial host. And so that seems just about ideal for a pathogen, for example. The reality is these have been around for a long time, since the early part of the last century. They have been used for therapy, for sure, but they were sort of displaced as antibiotics came on the scene for a variety of reasons. With resistance, we're now looking at phage and bacteriophage parts as other ways to treat infectious diseases that are life-threatening primarily. There have been multiple usages that have been reported. Many are, or actually all of them, I believe, are compassionate use. And so these are sort of one-off trials of phage for particular problems. And there's some great literature on the use of phage to treat mycobacterial infections that can be drug resistant. They are precise in that they're usually species specific, sometimes strain specific. They can be very effective, but there are challenges along the way. The bacterial phage arms race has been going on for 3.5 billion years. And so both sides have a substantial armamentarium that's been developed, and bacteria still exist along with phage. And so they don't kill all of their hosts, obviously. And there are various reasons for that. So resistance in various forms becomes something that's very important for phage. They can be relatively easy to produce in some cases. In other cases, they require growing large amounts of a pathogen to produce, and that may or may not be problematic. But what they do, which is usually a good thing, is they kill bacteria through mechanisms that are completely different from antibiotics. And so antimicrobial resistance is often not a problem for a phage to kill a bacterium. In the cases we've seen that are successful, some are not, many are, they're sort of one-off therapeutics. And there is an extensive amount of personalization or precision that's actually required. And in some cases, you take an isolate, for example, a Mycobacterium abscessans from someone that has cystic fibrosis, and screen large numbers of phage to find those phage that are just right for killing that bacterial cell. And so if we're going to see this blossom into a general strategy that is economically viable, we need new types of sort of diagnostics and ways to very quickly, using high throughput methodologies, pair bacterial pathogens with phage that may be efficacious. I think we're in the very early stages of this. We're finally starting to see a lot of clinical trials. The research community is very much engaged. There's a lot of really interesting work. And there are also parts of phage, which maybe we can talk about a little bit later on, that can be effective and actually have some advantages over complete phage. But as a microbiologist, it's hard not to get excited about what's happening in this area right now. And I think maybe Dr. Craig would like to comment as well.
Yeah, John. Yeah, I would love to hear from you. Does the CDC think of this as a potential general strategy?
We think there's an incredible amount of promise with phage, and I'll just note there's a couple other applications of phage that we've looked at, and that I think others are looking at too. Is that they can be also you can potentially impregnate them onto things like a catheter or onto potential surfaces to try and have antimicrobial properties against those for prevention purposes. So we think there's a lot of potential, and I think this is what we would flag for everybody. And I think that thanks to Research for America for holding this meeting and talking about this, because I think that we do need greater investment. We need greater research on these types of modalities. And I think the other thing that I would highlight is that at the end of August 30th, CDC and FDA actually held a meeting about alternatives that are preventatives for antimicrobial resistance. So it's not necessarily just the antibiotic drug pipeline that we need, but also the preventative agents. So how can you, to Jeff's point, have agents, and some of them could actually be phages, that could decolonize a person? So we carry some of these germs in our guts, on our skin. cystic fibrosis patients can carry them in their lungs, and that colonization with those resistant pathogens can lead to an infection and can importantly transmit that pathogen more widely in a community or in a nursing home or in all variety of cohorts of patients. And what CDC would like to do and would like to see is how can we get some of these decolonizing agents to market that don't create new pressures for resistance to develop, but can stop some of that chain of transmission and help those patients and prevent them from that colonization ever getting to an infection or an infection that could lead to their death or to have other serious consequences.
And Dr. Miller, you mentioned, this sometimes it's like a one-off therapeutic. Is this something that can be scaled up for broader use?
Yes, I think it can. And we're just starting to see the development of methods for this. We need large collections of phage with known host range determinants, et cetera. And so the fantasy, or maybe the future of having access to many different phage, a clinical isolate, and very rapid, almost point of care screening methodologies to try to identify the potential matches between which phage would be beneficial for which patients. There are other ways to approach this as well. For example, the phage injection machinery can be isolated as a tiny nano machine and programmed to penetrate bacterial surfaces, and that kills the bacterial cell. These are called tailocins. They have other names. They're naturally produced. Bacteria make these in nature. They can be exquisitely specific, rapidly reprogrammed, and they kill by generic methods that are really not susceptible to resistance as we know it. And so there are a lot of technologies that can be and are being developed to make this a reality. Phage license, the molecules phage use to destroy their hosts, can be exploited in isolation as well. And there are a lot of efforts to do that. To get back to what Dr. Craig was saying about decolonization and prophylaxis, the important point that he actually made is that if you have precision and you don't have to worry about selecting for transmissible resistance or decimating protective microbes, then the idea of using these entities prophylactically before someone goes into the ER, before someone goes into a hospital for a bone marrow transplant, as immunoablated and put on multiple antibiotics, this now becomes a reality. And so paired with the right kind of diagnostics, molecular diagnostics, microbiome-based diagnostics, for example, this may be something that we'll be looking at, not just to prevent Clostridium difficile infections, but to address other very acute infections that occur in the hospital setting.
And wanted to turn real quick to wastewater surveillance, which has kind of really come into the spotlight amid the COVID-19. Dr. Mathers, you've had experience at looking at wastewater surveillance for COVID-19 and AMR. So can you tell us kind of how this is being used to track antibiotic resistance and how common is using wastewater surveillance?
Yeah, thanks, Rachel. So, I would say writ large, antibiotic resistance wastewater surveillance is probably in its infancy, is how I would describe it. I think it's a really exciting approach. I've been doing, because we were seeing antibiotic resistant bacteria in our hospitalized patients and we could not figure out the source, we ended up going to premise plumbing, wastewater premise plumbing, and so developed a lot of techniques kind of from 2013 and on to really understand sort of the building microbiome, actually, if you can think about that. And then that building microbiome then gets excreted out into the wastewater. And so can we take advantage of that at various levels? And I think, like you pointed out, SARS-CoV taught us a lot about how we can use building level or wastewater level surveillance, community level wastewater surveillance to understand what's going on within the inhabitants of either a community or a building. And so, for example, we had used wastewater surveillance on building essentially for pooled testing to understand which buildings had patients with or without COVID, so we could go in and test all the individuals and isolate the people that were positive, and did it as sort of an early warning system for isolating people. You can either on campus and nursing homes where people live congregate in a congregate setting. Prisons could also work that way. But if you then take that knowledge and those learnings and flip it to what's going on in a hospital, then you could really inform infection control and prevention practices, and understand if there are emerging antibiotic resistant pathogens that you need to be on your toes about coming out of the back of a hospital. Also to understand what communities are potentially at high risk for antibiotic resistance. But what I would say, Rachel, is there's a lot of work to do, because there's a lot of genes of drug resistance in wastewater, and they're not all clinically relevant. And so we need to decide and really develop some methods and come up with some rules of the road about how we're going to do this at a molecular and then apply it for an epidemiologic basis. But it's a really exciting time for this technology.
And just as a quick reminder, if anyone has questions, please put them in the Q&A box, as we have about like 15 minutes left of our discussion. But Michael Crick wanted to bring you in. Is the CDC investing in wastewater surveillance, not just for COVID-19, but for AMR as well?
Absolutely. And we're so fortunate to have Dr. Mathers on, because she is one of the leaders in this field, and we're excited to have always worked with her for a number of years, and this is taking off, as she notes. And I'll in fact note to you that not only are we looking at it for antimicrobial resistance, but in fact, we were looking at it for antimicrobial resistance prior to the pandemic. And in fact, the work that we were funded, and I think this is one of those great stories about the importance of research that you never quite know what's going to come of it, is that we funded a antimicrobial resistance project at a wastewater treatment plant to literally identify AR pathogens before the pandemic and see what the risk was to those to the workers in there of getting an AR pathogen. And what happened is then the pandemic set upon us, and that project actually became the basis for detecting COVID. So the methodology of collecting it from the water treatment plants, the methodology of all of the we had sort of done to come up with a good method of collecting it, and what was the interval of testing, et cetera, all of that pivoted then to COVID-19. And so that method was then used for our national wastewater surveillance system, or NEWS, which is now detecting COVID. And so now we're actually at the point where now that we've stood up this surveillance system for COVID using wastewater, we're now going to try and pivot back and look at AR pathogens. And so going back to those, and Dr. Mathers, I think, gave a great insight that we're looking at it both from what can we see what's happening at a healthcare institution, whether that's a hospital or a nursing home, what pathogens are there potentially not being detected that we need to go back and screen for. And then we're looking at it at a community and seeing what is going on across that community, whether there are pathogens in that community that are circulating that we actually haven't identified previously, whether there are areas of concern that we didn't know about previously. And I think the other application is that I'm keen on looking at is globally, this, I think, has a lot of potential for countries that don't have mature AR surveillance platforms. And that's an area that we really haven't touched on a lot here, but the global burden of AR is much larger than any of us appreciated. The January Lancet article noted that it's larger than HIV and malaria, and that this is a tool that we could see being very powerful for helping some of those low to middle income countries identify areas of concern in their countries, and potentially deploy interventions to address those.
And we have a question in the chat about precision antibiotics. So Dr. Miller, if you could take this. How can precision antibiotics overcome the challenge of rapidly mutating bacterial targets? Is there a shift towards developing vaccines against drug-resistant infections versus developing therapeutics that are less marketable?
A great question. resistance is always a problem with any antimicrobial. And I think one of the questions are, a few of the questions are, number one, are there any evolutionary constraints that might limit that? And certainly we see with some bacteriophage that they tend to recognize receptors on bacteria that are often relatively immutable. And that turns out to be useful as a means of deciding how to target, say, a recombinant entity or recombinant phage or phage-derived entity against a particular pathogen. So resistance is a problem. Reprogrammability of the agent, which we typically don't have, at least in a facile way with most antibiotics, is important. The issue of vaccines is really important. As we, I think, all have learned from SARS-CoV-2, prevention of an infectious disease is much more effective than therapy. When we prevent an infectious disease, we're dealing with fewer numbers of pathogens. The ability to escape vaccines for most pathogens is less than the ability to escape a therapeutic. SARS-CoV-2 happens to be unexpectedly proficient at escaping immune responses. But that's not necessarily true for all the bacteria for which we have vaccines. With the pneumococcal vaccines, for example, particularly the new ones, we see a decrease in antimicrobial resistance in pneumococci. And that has an effect that is important for those who aren't vaccinated or have not been vaccinated against the pneumococcus. And so there are a few things better than a really good, safe, efficacious bacterial vaccine, certainly in terms of antimicrobial resistance. I think the bottom line is with the dearth of new antibiotics, truly new entities that are coming to the pipeline, which is becoming increasingly problematic, although there is an uptick in recent years, we need to be looking at every potential therapeutic and preventative solution that we can come up with.
And Michael Ybarra, yep, I was going to see if you could weigh in on this for us.
Well, Rachel, one thing I just wanted to add there on the power of vaccines for AR pathogens is something that goes incredibly underestimated, but it's incredibly important. And that is vaccines, regardless of whether they're bacterial vaccines or viral vaccines, tend to reduce antibiotic use, and they can reduce it pretty substantially. We shouldn't see them reduce antibiotic use really for a viral vaccine, but we also know there's a lot of inappropriate antibiotic use. We know that still antibiotics are commonly prescribed for COVID-19. They're commonly prescribed for flu. So the more we vaccinate those, the more we vaccinate folks against those viral pathogens, the less that there's going to be of that inappropriate antibiotic use and the less demand. Same too for all of the things Jeff noted for the bacterial pathogens as well. This is a really powerful antibiotic stewardship tool, and we need to talk about vaccines more and more as one of our most powerful tools for antibiotic stewardship.
Yeah, and Michael Ybarra, I was wondering if you could weigh in on the question of, is there a shift towards developing vaccines against drug-resistant infections versus developing therapeutics that are less marketable?
Yeah, no, it's a great question. I mean, a couple of things jumped out in the last few minutes to me, and I'll just give a quick clinical example where, like Dr. Mather, some of the most dramatic cases that I've seen are people, even young people, college students. I work at an academic medical center on a college campus, and young kids coming in, or young adults that are college age coming in with sort of severe bacterial infections, things that you don't think about as a parent, but you worry about when sending your kids to college, some of which are actually vaccine preventable. Where vaccines can prevent infection for pneumococcus, for example, or other bacterial infections. So vaccines play a really important role here. The other component of this was diagnostics, which Jeff Miller was mentioning. Diagnostics are really important because when I see a patient right now, if I swab something or if I send something to the lab from a patient, what I end up doing is checking on the lab 1, 2 days later, 3 days later, seeing what antibiotics it was resistant to. So the development of diagnostics is also really important. So at the point of care, I can sort of determine, is this a run-of-the-mill bacterial infection? Is it a viral infection? Or is it something more serious? And then third, it's really all of the above. We need all of the solutions that were mentioned, these novel technologies. But as Dr. Mathers mentioned, we also need sort of antibiotics, the traditional antibiotics, so that when somebody's hospitalized or in the intensive care unit, you have that antibiotic to give to the patient that may be crashing or super sick because they have a multi-drug resistant infection. The solutions that we're advocating for are all of the above. What some of the what I think the Pasteur Act would do is really focus on, again, highly novel antimicrobial agents, both against treatments for bacteria, but then also fungus as well. And so I just all of the above plus the stewardship is really what we need to get at this infection. And I just appreciate all of the scientific knowledge that I've gained on phages as well from Jeff Miller. So really, really great discussion.
And as a reminder, we have just about 5 minutes. So if you have any questions, feel free to put them in the Q&A chat. But it looks like right now, just wanted to, as we start to close up, get thoughts from kind of each of the panelists on kind of what's sort of we've talked about a lot here. So kind of just in summary, what is your kind of biggest question or one sort of potential solution to beginning to turn the tide on AMR that you see, just kind of in a brief minute or two here? Dr. Miller, we can start with you.
Sure, thank you. As an academic and an entrepreneur, what concerns me most is not so much a state of research or a state of knowledge, it's the business model. And if you look again at the innovative startup company environment, it is an acute problem. And I know that there is a little bit more interest in big pharma, but leaving this up to that marketplace has put us in many ways in the situation we're in, and we need to fix that. And so I think if I were to have one request for the policymakers, it would be to help incentivize antibiotic development and deployment, and to keep the innovative pipeline not only functioning, but invigorated for developing new modalities.
Michael Craig, let's go to you.
Yeah, I think I would just reiterate what I noted before, is that we would really like to see not only the antibiotics invigorated, but also the preventatives. So more that we can do to bring vaccines to market. As we noted, the power of the vaccine, not only to prevent the infection, but reduce the antibiotic use. And then the preventatives that we talked about, and these could be preventatives like that use phage. They could be preventatives that help support the microbiome, but they're going to take care of that colonization with these pathogens. They're going to reduce the transmission of it, which we see is increasingly a major driver of this, especially in healthcare settings, but even beyond that. And if we can bring some of those to market and they're successful, they could be an incredibly powerful tool in our public health and clinical armamentarium to really fight antibiotic resistance wherever we find it.
Dr. Mathers?
Yeah, so I think, my final thoughts are, as we're emerging out of this pretty tough pandemic for healthcare, it's really nice to see just the data coming out about what really happened. I mean, on the ground, I could see antibiotic resistance emerging in our hospital and misuse of antibiotics, and everybody was stretched too thin to really get ahead of it. And so I think coming out of that, really thinking creatively about how do we take this opportunity to re-educate people about the use of antibiotics and how to judiciously use antibiotics and get people on board. And then also from a, antibiotic surveillance and resistance modality, again, labs were also spread very thin, and CDC knows that people were not actually submitting as many antibiotic resistance profiles. And so there's a big data gap right now in the United States about how much resistance is actually going on because labs were spread so thin. And so creatively using tools like wastewater to improve the passive wastewater or passive antibiotic resistance so that we can respond to it in a more resource thoughtful way. And then, of course, I would just echo what others are saying about, I can't emphasize enough how much we need antibiotic development, and agree with all the other panelists about forward-thinking prevention and drugs on the other end to treat infections. So thank you, Rachel.
And Dr. Michael Ybarra, we have just about a minute here, so up to you.
Yeah, well, thank you all. So thanks to Research America and all the great comments from the other panelists. I think, we always tend to be afraid of things that are sort of unknown, like what's the next novel virus that we don't yet know about. And yet this is one that we sort of know the problem. It's right in front of us. It's very personal to me in particular, taking care of patients. And again, some of the hardest cases that I've taken care of are really severe infections that you might assume could be treatable. And unfortunately, in some situations, they're not. So we know also the solutions. And I think there are very few things in healthcare where everyone agrees that we need a solution here. And there's actually generally some agreement on what that could be. Whether it's, collaboration like we saw in COVID-19 between private sector and the government, the focus on the problem, I think the next step is to move to action. And that's why I keep reiterating the Best Year Act is one of those solutions. And hopefully Congress will make a move this year. So thanks for putting it together and appreciate the comments.
And thanks to all our panelists and for everybody listening in the audience. Hope everyone has a good afternoon.